returning back to the NIH in 2000. In 2010, I came back to Children's part-time in my semi-retirement to direct what we now call the Grants Enhancement Program. So today we're very fortunate to have our speaker for the Jill Joseph Endowed Lectureship, Tom Boyce. Having worked with both of these special people, I was given the honor of introducing today's lectureship and our speaker. First, a word about the namesake of the lectureship, Jill Joseph. Often we have a picture of the namesake for the lectureship, but today it's not necessary because Jill is here in person. And Jill, could you stand up, please, and let everybody get to know you. In 1997, we recruited Jill to come to Children's to direct the then Center for Health Services and Clinical Research that is now known as the Center for Translational Research. As a seasoned, productive, and respected mid-career PhD behavioral epidemiologist, Jill had realized the value of medical training for her research and returned to the classroom for an MD and then a pediatric residency. So, as a strong hospital-based pediatrician, in addition to her own outstanding research in health disparities, supported by a series of NIH R01 and other funding, Jill provided leadership to build a strong center with the crowning accomplishment of winning the prestigious CTSA, the NIH CTSA award as the PI, an award that is now renewed under the leadership of Lisa J. Woodford. Now, relative to this slide that you see, with Jill's leadership and input, a collaborative center for health disparities has now been awarded for 15 years. And this is a partnership of Johns Hopkins, Howard, and Children's, funded by NIH to support research, training, and outreach to address health disparities. It also supports the Health Disparities Interest Group, and for any of you who may be interested, participation in that group is available through the connection shown on the slide. Beginning in 2002, Jill occupied the Agnes Hudson Endowed Chair for Health Services and Community Research. However, following family concerns and opportunity, in 2011, Jill returned to California to become the Associate Dean for Research at the Betty Irene Moore School of Nursing at UC Davis. Not just an ordinary school, a new, innovative graduate nursing school funded by a $100 million grant from the Gordon and Betty Moore Foundation, the Intel co-founders, to improve health and reshape health systems through innovative research, education, and advocacy. Based on the impact of Jill's administrative leadership, her excellence as a mentor, and her research accomplishments, upon her departure, this lectureship was established in her honor. In introducing our speaker, Tom Boyce, I faced a serious dilemma. There was so much to tell about him that in the telling, I would regrettably detract from the opportunity for you to have the opportunity to hear directly from him. Therefore, I must be far shorter than I would like or than Tom deserves. Very quickly, his academic demographics are bachelor's at Stanford, MD from Baylor, pediatrics at UCSF, Robert Wood Johnson Clinical Scholar in Pediatrics and Epidemiology at the University of North Carolina, Assistant to Associate Professor at the University of Arizona, Associate to Full Professor at UCSF, Visiting Research Scientist at the renowned Primate Library of Comparative Ethology at NICHD, Dean for Research and Head of Community Health and Human Development at UC Berkeley School of Public Health, Endowed Chair in Child Development, University of British Columbia, Vancouver, and currently the Pritzker Distinguished Professor of Developmental and Behavioral Health at UCSF. He has over 150, considerably over 150 original peer-reviewed publications, 19 book chapters, 3 books, and virtually pages of awards and honors. Tom has evolved as a special kind of developmental and behavioral pediatrician and researcher. His focus has been on environmental, biological, and genetic basis and contributions to the developmental and behavioral vulnerabilities and morbidities of children. Yes, as evidenced by such titles as Crowding Stress and Violent Injuries Among Behaviorally Inhibited Rhesus Macaques, The Confluence of Childhood Mental, Physical, Social, and Academic Difficulties in Middle Childhood, Exploring the Headwaters of Early Life Morbidities, Development in the Epigenome, the Synapse of Gene-Environment Interplay. Though Tom tells me that his primary qualifications are being a father and grandfather, he is internationally recognized as an insightful, creative, and cutting-edge thinker and investigator. For the junior faculty and the fellows, as you will see over the next hour, when you're at the PAS or some other similar academic meeting, you won't want to miss the opportunity to hear about what he's up to and what he has to say. So please welcome our Jill Joseph Memorial Speaker, Tom Boyce.
Thank you, Peter, for the kind introduction. Peter actually was my program officer when I got my first R01 from NICHD many moons ago, and we've been good friends since. Tam Hines was a colleague of mine at the University of Arizona, and I've known Jill since both of our days at University of California, Berkeley. I've wondered whether I was supposed to sing the lecture, given that he's standing over in the corner, but I will spare you that. I am honored to be here giving a lecture in Jill's name, and as you know, this is one of the premier pediatric institutions in the country, so I'm also honored to be here at Children's National Hospital. The title of my talk that I chose for today was Gene-Environment Interplay and the Biology of Misfortune. My work in this area really began driven by this phenomenon that is the single most well-replicated finding in all of child health services research. And that is that within any given population of children, there's a group of about 15 to 20%, about 1 in 5 children represented by these darker figures, who are responsible for well over half of the biomedical and psychiatric morbidities within that population, and are also responsible for well over half of the healthcare utilization. This tremendous non-random distribution of morbidity within childhood populations. The same is true of adulthood, and many of these individuals that are disproportionately affected by various pediatric morbidities in childhood go on to those who are similarly affected in adulthood. The public health ramifications of this finding are probably obvious, that if we could figure out what was going on with this subset of children, we would be potentially addressing well over half of the morbidity within the childhood population. So a compelling, uneven distribution of morbidity that right from the beginning of my career felt like an important kind of problem to solve. So this was the kind of professional motivation for the work that I've done over the years, but there is a more personal one, which in my dotage I have begun to share as well, and I want to tell you about that as well. So this is a picture of me and my sister Mary, me at about age 7, Mary at about age 5. And we were happy little kids growing up in Southern California. One of these individuals has had a life of almost shameful good fortune, a gratifying and productive professional career, has a stable 43-year marriage, has 2 thriving children, 2 grandchildren, and 2 more on the way, and few or no disturbances in health. But by contrast to that, my sister Mary had a life of real disappointment and affliction. She developed a disabling chronic disease at age 11. She carried a diagnosis of schizophrenia by age 20. She had an unplanned pregnancy and delivered a disabled child who is a delightful young adult with special needs, now age 38. and she committed suicide at 53 years of age. So the personal aspect of this uneven trajectory of health and development in two individual lives stems from my own personal experience in my own family. And the questions that have always gnawed on me are those having to do with why and how. Why are two children born into and raised in the same family? Why do they have such tremendously different trajectories of health and development? I think of this as being basically the twin human mysteries of asking why some get sick and others do not, and what is the origin of these kinds of differences that we see within populations of children. So I want to tell you today a story in three parts. The first is the story of how early socially stratified exposures to adversity and stress become biologically embedded, actually embedded within the physiology and biology of individual children, affecting developmental and disease risk over the entire life course. Second part of the story is that such risk is highly variable from child to child and is influenced by even the most proximate, immediate experiences of social subordination. And the third part is the observation that genes and social environments appear increasingly to operate together to produce these powerful developmental effects. So three parts to the story, and I will wade into that now. So this begins, one of the explanations for this extremely uneven distribution of morbidity within childhood populations is the one represented by this sort of iconic inverse association between socioeconomic status going from low to high and the percentage of children with any limiting condition, ear disease, physical inactivity, asthma, and injury. This highly replicable, found in virtually all childhood populations in which it's been addressed, a kind of social partitioning of child health and development. This is equally true, by the way, of mental health outcomes. This just shows for different age groups the same graded association between the level of poverty experienced by a child and the percent with emotional or behavioral problems. So poorer children sustain higher rates of acute and chronic physical and mental disorders, school failure, and delinquency. This is not simply an effect of poverty. There is a graded linear association with higher rates of disorder at every step of the social class gradient. Part of the mystery of this gradient is why it is that children at this level of wealth and well-being have slightly higher and significantly higher levels of these morbidities than children who are from the highest tiers of socioeconomic status. This renders SES the single most powerful determinant that we know of health outcomes, and yet it is a determinant that we only began really studying in its own right about 20, 25 years ago. So this very powerful association that we really have a relatively rudimentary understanding of. And there are various explanations that have been proffered for why this social class gradient exists, and all of these are legitimate. There is differential exposure to toxins among kids growing up in poverty and disadvantaged communities. There are differences in diet and access to health care, housing, exposures to violence in the family and the community, differences in parenting and parenting style. But the one that I want to focus your attention on today, which I think is one of the most salient differences, is the differences in exposure to adversity and stress among children from disadvantaged communities. This is what this looks like. This is from the work of Gary Evans at Cornell. When you look at children growing up in impoverished circumstances versus those in middle-income circumstances, the level of housing density, other housing problems, ambient noise in the physical environment, family turmoil and chaos, and family and community violence is substantially higher among children growing up in disadvantaged relative to middle-class families. This has consequences biologically. This is the work of Andrea Denise from London, showing how child maltreatment predicts adult inflammation. This is C-reactive protein, white cell counts, This one is inflammation factor, and this is fibrinogen. Each of them a graded association with levels of child maltreatment experienced during the childhood of these individual adults. This shows the childhood experiences of adversity and trauma predicting leading causes of adult mortality. This is the now famous work on adverse childhood events done by Vince Felitti and his colleagues in San Diego, Kaiser, and the CDC. For each of these adult causes of mortality, coronary heart disease, diabetes, sexually transmitted disease, drug, and suicide, the higher the levels of these adverse childhood events, the higher the levels of these predictors of adult mortality. It's also true that these kinds of adversities, both the acute childhood adversities studied by Felitti as well as more chronic ongoing adversities, have a very high attributable risk for psychiatric disorders. This is from the work of Katie McLaughlin. She shows that when you look at population attributable risk, which I'll remind you is the level in the exposed individuals minus the incidence in the unexposed, essentially the reduction in incidence that would be observed if the population were entirely unexposed compared to its current exposure pattern, and you look at a variety of psychiatric disorders, the fear subclass, distress subclass, behavior, substance abuse subclass, you can see that there is a substantial population attributable risk, and overall nearly 30% of adolescent psychopathology is attributable to these kinds of adversities. So very prevalent, very disproportionately represented within and unevenly distributed within communities of poverty and disadvantage versus middle-class communities, and a very large portion of psychopathology and other forms of morbidity attributable to these kinds of adversities. So it turns out that there are just these pervasive differences in the experiences of everyday life between children who grow up in disadvantaged circumstances and those who grow up in middle-class environments. But it is also true, and this is another version of this same gradient. This is showing social class background and reading performance. Here are these nice linear associations by country. When you look at the individual data points, as in this relationship between social class and receptive vocabulary in Canadian children, there's a tremendous amount of variability. This is a very powerful linear gradient, highly unlikely to be due to chance, and yet it looks like a shotgun hit the screen here. There's a tremendous amount of variation in the tightness and the power of this association. Similarly, ACEs scores predicting lifetime histories of depressive illness. There is this, again, linear gradient as you proceed up. But when you look at the actual data points, there's far more variation in this association than you would suspect based on these kinds of presentations. So so stressful and disadvantaged environments seem to have these highly variable effects on developmental and health outcomes. We struggled for a number of years in trying to take the noise out of these associations using stronger and stronger study designs, prospective looks at this association, using better and better measures of both developmental outcomes and measures of stress and adversity. But after a while, we began to wonder whether the noise was not actually the music and actually the phenomenon that we needed to be looking at. This variability in children's responsivity to the stressors and adversities that they experience. So about 25 years ago, my nursing colleague, Abby Alcon, and I began bringing children into these laboratory situations where they were seated in front of a previously unknown examiner. They were presented with a series of ecologically cogent and appropriate challenges, just stressful enough to evoke changes in their biology, but not so stressful that they ran out of the room. And it took us a while to figure out where that line was. And as we did this, we monitored the two principal stress response systems in the human central nervous system, the hypothalamic pituitary adrenocortical system, or the corticotropin-releasing hormone system that's centered here in the hypothalamus and the arcuate and paraventricular nuclei, and the locus coeruleus norepinephrine system centered here in this little nodule in the brainstem, which is responsible through dopaminergic projections to the hypothalamus for igniting the whole fight or flight response that we associate with the autonomic nervous system. So, adrenocortical and autonomic reactivity to standardized laboratory stressors, very scripted, very standardized experiences. We measured salivary cortisol responses. We measured impedance cardiography for assessing the fight or flight response within the autonomic system. And what we found was that there was very broad and very reliable individual variation in the magnitude and patterns of the responses that these children showed us. We then were able to take these very standardized measures of stress reactivity and bring them into the naturally occurring world that the children were actually experiencing, and now relating naturally occurring stressors to psychopathology and other kinds of pediatric morbidities, but divided into those who were high and low in this stress reactivity in these two principal systems. So here is one representative finding looking at pre-syndromal psychopathology as a function of the level of family conflict, high versus low, and the sample is going to be divided into those who are the lowest in ANS reactivity. That's about 80% of the children within our samples, versus those who are high in ANS reactivity. And here's what we found. We found first that the children who showed us very little ANS response in the laboratory, they were relatively indifferent to the effect of this naturally occurring family conflict. They had a modest increase in pre-syndromal psychopathologic symptoms, but really none to speak of at all. By contrast to that, the children who had high ANS reactivity in the lab had either the worst outcomes or the best outcomes, depending upon the character of the family environment in which they were being reared. The kids being reared in families with high marital and family conflict had the highest level of symptoms. Those being reared in low family conflict environments had even lower levels of pre-syndromal psychopathologic symptoms than their peers who were low in reactivity. Years ago, when I first began presenting these kinds of findings, I was presenting at Stanford, and this old guy came down after the talk with a cane, and he pointed the cane at me, and he said in a thick Swedish accent, "You're talking about Maskrosbarn." And I said, "I don't know what Maskrosbarn is." And he informed me that Maskrosbarn is the Swedish idiom for dandelion child. It literally means dandelion child. And what the Swedes mean by that is a child who is able to grow anywhere, like the dandelion. The dandelion grows in the cracks in a brick wall on the streets, the cracks in the sidewalk in a fertile mountain field. So those are the dandelion children that show this very almost no response to high family stress and other stressor exposures. We, of course, coined then the countervailing Swedish neologism, which is orchidbarn, orchidebarn, for the orchid children who, like the orchid, can flower into exceptional abilities, exceptional outcomes under good conditions, but have tremendous vulnerability to not thriving under the worst of conditions. So these children turned out to be those who are neophobic. They tend to withdraw from novelty and challenge. They are kids who are disproportionately shy in temperament, and they are reactive within the laboratory and show a variety of sensory kinds of sensitivity. So here was evidence of what we have since called differential neurobiologic susceptibility to social context. Notice that it is not simply that there is a group of vulnerable children and a group of resilient children. It is rather that there are vast differences in the sensitivity of children to the social environments that they are experiencing on a day-to-day, week-to-week basis. We found the same thing in a troop of rhesus macaques studied not far from here in Poolesville, Maryland. This is a troop of animals that live on a natural enclosure, a natural habitat that has a pond and trees to swing from and lots of toys, sort of like a summer camp for monkeys. And we happened to be there immediately following a period of confinement when all of these animals that had been living in this wonderful 6 or 8 acre enclosure were having to be confined to a 200 square foot cinder block building during a period of reconstruction on the natural habitat. This was, of course, extremely stressful to this troop of animals, and they had been previously characterized as being high or low in reactivity in a variety of ways, mimicking very much the kind of work that we had done with children. So we were able to look at these individuals in a low stress year and a confinement year and look at the effects of those exposures on violent injuries sustained during those periods of time. And here is what we found. Basically the same finding, but in rhesus macaques, that there is a vast group of low reactivity individuals who essentially had no increase in violent injuries under the confinement stress, and a smaller group of very reactive monkeys who had high levels of violent stress. In fact, one of them died during the confinement period versus very low rates of injury during the pre-stress year. So here again, the dandelion macaques and the orchid macaques, using that metaphor again. We have also found this at the level of allelic variation. For example, Nikki Bush, one of my past postdocs, looking at fall chronic cortisol secretion levels as a function of family income, and looking at the net carrier allele versus the Val homozygous allele in the BDNF gene, the brain-derived neurotrophic factor gene. Again, the same phenomenon of there being orchid individuals who had either the highest cortisol levels or the lowest, depending upon socioeconomic status, versus the majority of children within this cohort who had very little change in cortisol under the different family income circumstances. It turns out that this is also a phenomenon that not only affects morbidities and outcomes, but also affects kind of normative developmental processes. Children, if we look at pubertal development going from Tanner 1 to Tanner 5, we all start here and we all end up here. This is age centered on 12.5 years, going back 3 years and forward 3 years. So basically the course of puberty. And we divide this into children who had low sympathetic nervous system reactivity. These are going to be shown in blue, versus those with high sympathetic nervous system reactivity. And they're further divided into those who had parents who were very low or high in their warmth in a parent teaching task years before the onset of puberty. And here again, we find the same kind of thing, sort of a linear progression of puberty in the children with low sympathetic reactivity, but in the children with high sympathetic reactivity, either the most rapid progression of puberty, which, as you may know, is associated with higher risk for pregnancy, for acquisition of an STD, and for a variety of outcomes that we would like to avoid, versus those children who had high warmth parents and high sympathetic nervous system reactivity, who basically didn't even go into puberty until 12.5 and then caught up with the rest. So here again, the phenomenon of the dandelions and the orchids, who have either the worst of the outcomes developmentally or the best, depending upon the character of the parent-child milieu in which they are being raised. So this phenomenon of differential neurobiologic susceptibility to social context we have now studied at a variety of levels, ranging from the society surrounding children through behavior. We've studied it at the level of brain, of circuitry and synapse, and we are now engaged in studying it at the level of the genome and the epigenome. So as I head now into the second part of this story, I want to tell you briefly about a study that we did of socioeconomic status, stress, and oral health. Why study dental caries? It's the single most common chronic disease of childhood. There are strong socioeconomic and racial disparities. We know some of the etiologic factors having to do with lead exposure, tobacco smoke, diet, and so on. We know that there are bacteria that are implicated, the so-called cariogenic bacteria of strep mutans and lactobacillus acidophilus. And the leading account of these disparities in dental hygiene and oral health is that the low SES parents are neglecting the dental hygiene of their children. We didn't believe that that was an adequate explanation, and so we began to pursue this in more detail. In these studies of preschool and early primary school children, one of the things that we have wanted to capture was their circadian cortisol secretion among kids of different socioeconomic status. And we had been relegated to the relatively inadequate sampling of cortisol in saliva at 3 different points during the day. It's a snapshot of cortisol secretion at different points over the circadian cycle, but what we really wanted was the area under the curve. We wanted to know this continuous secretion of cortisol over the period of from sleeping to waking. So we pondered over and over again about how we could, what would be a biological measure that would allow us to assay cortisol secretion in a more continuous area under the curve manner. We thought about Pott's disease, which of course is not Pott's disease, Cushing's disease, which of course has osteopenia and vertebral collapse as a consequence of ongoing high cortisol secretion. We obviously weren't going to x-ray all of these children that we were studying in normative childhood conditions. But then we realized that the kids that we were actually studying were going to be losing a calcified tissue the next year following our study. They were going to begin exfoliating their primary dentition, which of course is a calcified tissue that in animal studies has been shown to be also responsive to the effects of cortisol secretion in a manner similar to the osteopenia that we see in Cushing's. So we asked the question, might deciduous teeth function as a stress biomarker in young children? We thought that perhaps the different compartments of the microanatomy of the tooth, or the density or the solidity of the enamel layer might be affected by cortisol secretion. We used micro-computed tomography to measure both the size of the compartments of different compartments of the microanatomy of these exfoliated teeth, and we also used CT to measure the solidity and hardness of the dental enamel layer. I should tell you this was called the Tooth Fairy Project, of course, and we offered $10 per tooth, which was substantially above market rate at the time. So we harvested an awful lot of deciduous teeth from these 6-year-old children. And in fact, we got one tooth that we sent over to our colleagues at the dental school at UCSF, and it came back with a Post-it on the bag that said, "This is a dog's tooth." So somewhere in Berkeley, there's a dog that's kind of snaggletooth and with an owner that is $10 richer. What we found in this study, I won't bore you with the details of the actual data, but we basically found that there were these sociobiological effects on childhood dental caries, and the linkage between socioeconomic status and dental caries was actually subsumed by 2 different pathways. One was an earlier and more florid acquisition of cariogenic bacteria in kids from low SES families. The other was the presence of financial stressors in the lives of these families, which led to greater HPA activation in their children, which resulted in changes in the dental microanatomy that essentially rendered these teeth more vulnerable to the effects of the cariogenic bacteria. And in fact, the strongest predictor was the interaction effect between bacteria and cortisol in predicting dental carie incidence. So oral health disparities related to two interacting pathways, earlier and more intensive exposure to an agent of disease among low SES children, and more family adversity resulting in greater HPA activation, cortisol secretion, and microanatomic vulnerability. This led us, however, as we began to study this in children as they gathered into new kindergarten groups coming together, whether there might be aspects of this social gradient in outcomes of various morbidities beyond the kind of visible surface forces that drive socioeconomic health disparities. In other words, factors that might go beyond toxins, diet, healthcare, housing, violence, diversity, and stress affecting these little individuals afloat on a vast ocean. And there might be these deeper, less visible currents of social inequality that actually occur within micro-societies of human children. In other words, the question we were asking was, over and beyond the effects of socioeconomic gradients on morbidities in children, is there an effect of the social inequalities that we were observing in small groups of children coming together for the first time in kindergarten classrooms? We know that African cichlid fish maintain severe hierarchical organizations in which only the dominant males have reproductive access to females. We know that rats in a subordination paradigm show pro-inflammatory shifts in cytokine signaling pathways. We know in primates that they form these stable, linearly transitive social hierarchies, and that subordinate positions within those hierarchies are associated with upregulated adrenocortical function, impaired immune competence, and decreased resistance to disease. We also know and observed and knew from the work of other social scientists that young children form these social orders within weeks of entering new social groups. My medical students that I teach this to hate this fact, that there are these gradients that groups of 20 and 30 children form, but you ask any kindergarten teacher and he or she will tell you that they can assign the pecking order of each child within 2 to 3 weeks of the inception of a new class. So the question we asked was whether these subordinate positions, deeper current in early peer hierarchies, was associated with greater stress, exaggerated reactivity, and excessive stress-related morbidity. We used a naturalistic measure of dominance and subordination behavior in groups of children. We defined social dominance as a pattern of repeated encounters in which the outcome consistently favors the same dyad member. We did dominance observations where we did critical event and scan sampling over 3 to 5 weeks of time within kindergarten classrooms, and the behaviors that we looked at were things like physical attack, imitation, directing, threat, relational aggression, all things that had to do with dominance and subordination. And having done that, we could then assign children within a classroom to a position in this linearly transitive hierarchy. I was going to show you this video of the behaviors, but I think I want to skip that so that I can get to the meat of what I'd like to convey. So the question really was, we know that this is true within nations. The question was, within 338 kindergarten children in 29 Berkeley public school classrooms, would the same be true? And it turns out that it is true. Here is social position among boys and girls, predicting depression levels, peer relationships, inattention, academic competence. And what we found is shown up here, that subordinate children sustain higher rates of depression and inattention, poor peer relationships, and academic competence. And these tended to be associations that were steeper and more reliable in the boys than in the girls. We also studied the potential effects of these individuals who are the leaders, the teachers, who I am now convinced we pay way less than what we ought to be paying them to ensure the well-being and the future of our children. Having a good teacher in kindergarten completely ablates those associations with depression and peer relationships and so on. The teachers have this powerful role of either enhancing or ablating these associations with social position. So powerful stress-related effects of social subordination itself and social marginality at both the societal and small group level, and effects that seem to be able to be mitigated by more egalitarian, empathic policies and practices within the classroom. So how does all this happen? We think that it happens now in the same way that these two phenotypes of butterflies happen. These look like two different species of butterfly, but in fact they are the same species of buckeye butterfly, and this is a polyphenism that is driven by temperature and length of daylight as they emerge from the pupal phase of development into their butterfly stage. So they are essentially sensing the character of the early environment and changing their phenotype based on those senses. So this is a conditional adaptation involving differential epigenetic regulation of the genes that determine coloration and pattern. For those of you who, just a quick refresher on what epigenetic marks actually are, if you unravel a chromosome into the chromatin that is contained within the chromosome, it is either tightly closed, which is hard to transcribe if you're an enzyme doing the transcription of a given gene, or it is in a loosely configured euchromatin format. All of the DNA is wrapped around these nucleosomes like beads on a string, and there are these chemical attachments on either the histone proteins or the DNA itself that shuttle between this open or easily transcribed chromatin and the closed, less easily transcribed chromatin. And it turns out that environmental exposures are much of what drives DNA methylation and the modification of these histone proteins in response to these kinds of environmental exposures, including those to adversity and stress. So the epigenetic processes serve as a kind of dimmer switch, if you will, that turn up or down the regulation of the expression of a given gene, and these epigenetic marks then constitute the physical nexus between genes and environments. We have had a chance to study this. We're now studying it in a variety of cohorts, but one of the early ones was within the Wisconsin study of families and work that began during the second trimester of pregnancy and followed children all the way through to their high school graduation. We were able to relate infancy and preschool parental stressors reported at the time of those stressors occurring and relating those to the epigenetic profiling of buccal epithelial cells in mid-adolescence. Here is what we found. We found that there were these epigenetic vestiges of early parental stressors. This shows those stressors during the infancy period and the preschool period for all children, girls and boys. The mother's stressors are shown in red and the father's in black. We found that the mother's stressors in the infancy period were more related to differences in methylation for both boys and girls. And the father's stressors in the preschool period were associated with demethylation differences primarily for girls. So patterns of association in these epigenetic profiles that are consistent with what we already know of the developmental time course of parental influence by gender. So when my first grandchild was born, I might have been asking this kind of a question: how do you want to play this, nature, nurture, or a bit of both? The answer is, of course, that it's both, always. And what we have come to realize is that we think that most of the developmental outcomes with which we are concerned as a society or as pediatricians stem from a combination of allelic variation in the genome, environmental variation in the character of the social world that the child inhabits, those being translated, if you will, through the epigenome and giving rise to adaptive and maladaptive phenotypes, or in the metaphor that I've conveyed to you, dandelion and orchid children. So the story in 3 parts has been one of socially stratified exposures to adversity becoming biologically embedded, this risk being highly variable from child to child and influenced by these very intimate, immediate experiences of social subordination, and genes and social environments operating together to produce powerful developmental effects. So the answers to my fundamental questions about my upbringing and my sister probably have to do with unshared family environment, raised basically in different families, although we inhabited the same home. Probably sibling differences in genomic risk, probably something having to do with birth order or gender, and probably, in all likelihood, complex epigenetic interactions between allelic variation and family environment. This was a note that was handed to me as a little girl, an 8-year-old girl, left the clinic a number of years ago. She had come to talk about her chronic abdominal pain. She shows these 3 big girls, and here she is, and down at the bottom she wrote, "I got left out when the big kids tease me, and my heart is like breaking." Here is Tim Kretschmer, a German secondary school student who wrote on the internet, "I am sick of this messy life, always the same. Everyone makes fun of me. No one recognizes my potential. I am serious." He posted this prior to a shooting rampage the next day, which killed 15 people. The most recent, perhaps generational manifestation of this is the Occupy Wall Street movement, which has risen up within a generation of young Americans as a protest against the kind of inequalities that they see within our American society. What all of these experiences have in common is social stratification, subordination, stress and adversity, and inequality in life chances. It is really what Pope Francis, I think, was talking about in his address to the UN General Assembly when he referred to a selfish and boundless thirst for power and material prosperity. We know that social relationships are important, and we know that they are particularly important in a society where income inequality is higher than it has been since the Great Depression. These are my collaborators that I love and appreciate, and I want to end with this one little story. This is a story that was taken, Max Dupree, who's the author of several books on leadership, wrote in his book, Leadership is an Art, about his granddaughter Zoe, who was born prematurely, weighed just a pound. She was in an NICU with multiple tubes and monitors when Dupree and his wife came for the first time to visit her. To complicate matters further, Zoe's father had jumped ship the month before she was born, and realizing this, a very wise neonatal nurse gave the grandfather his instructions, and she said this to him: "For the next several months, you are the surrogate father. I want you to come to the hospital every day to visit Zoe, and when you come, I would like you to rub her body and her legs and arms with the tip of your finger, and while you're caressing her, you should tell her over and over how much you love her, because she has to be able to connect your voice to your touch." So, friends and colleagues, we are living clearly in a time of widespread resurgent disregard, probably unprecedented within our lifetimes, for the care and protection of the most vulnerable and powerful of the world's people. In growing number of nations around the world, and perhaps most visibly and disturbingly in our own, the defenseless are bullied and mocked, the poor are blamed for their poverty, the homeless are scorned as slothful and inept, refugees from violence are turned away, the lowly are ignored, and those who are the least of these among us are rejected and forgotten. Most important for our concerns here is the reality that it is the children of our nation who are the very most vulnerable of those who lack power who are the least capable of standing alone without protection or help. I would argue that in such circumstances, I have become convinced it becomes really a moral imperative of pediatric medicine that we connect our voices to our touch and safeguard our society's most vulnerable, marginalized, and important assets, who are the children, who are our legacies and our collective future. Thanks.
Time for questions. Time for a couple of questions.
Management meetings coming next.
Roger.
That was fascinating. And one of the questions, as your work has evolved, is there differences that you see genetically or epigenetically between the two groups? You said the high and the low sensitivity reactors. How far have you progressed in that research to try to determine which are the most vulnerable in the population and maybe the ones that need to be pinpointed first before they develop the significant problem?
Yeah. So we and others have now begun to document epigenetic profiles that seem to be characteristic of kids growing up in impoverished, high adversity communities. That was the first one of those was the data that I showed you. We are just about to submit a manuscript which shows that the highly reactive children, as opposed to the minimally reactive, more resilient children, also have an epigenetic profile that is characteristic and unlikely to be due to chance, has survived all of the false discovery rates that you know are inherent in doing the multiple comparisons of many CPG sites in the genome. So we think that there is not only an epigenetic signature of early stress exposure, but also potentially an epigenetic signature to this phenomenon of stress sensitivity and sensitivity to the social environment. Interestingly, one of the genes in which many of these differential epigenetic marks have been located is a gene that codes for the structure of the face. And Jerry Kagan, the eminent Harvard psychologist, years ago made the observation, never really followed up, that shy children have narrower faces than children who are temperamentally more extroverted and less shy. So, you know, it's very early, a lot of work yet to be done, but it does look like there is going to be a signature for the highly reactive kids as well. Thank you all. Yeah.